સાકાર કરવા માટે અમારી દેવ દુર્લભ ટીમના અલગ અલગ સાથીઓ કામ કરી રહ્યા છે. લાઈફ સાયન્સ આ 32 વ્યાખ્યાન એ જ સાયન્સ ફેકલ્ટીનું મોટું પ્રદાન છે એ બતાવે છે. ટોટલ 125 વ્યાખ્યાન સુધી જાગૃતભાઈ નંબર આપી દીધા છે. અમારી યુનિવર્સિટીના રજિસ્ટ્રાર ડોક્ટર અધિર સોલંકી, અમારા સૌ ઈસી મેમ્બર્સ, વિવિધ સાયન્સ કોલેજના આચાર્યશ્રીઓ, અધ્યાપક, વિદ્યાર્થી ભાઈઓ અને બહેનો, વિદ્યાર્થીઓ માટે ગોવિંદ ગુરુ યુનિવર્સિટીનો આ પ્રકલ્પ સમગ્ર ગુજરાતમાં એને નવી એક ચિરોચિત્રનું કામ કર્યું છે. આજના વિદ્વાન વક્તા ડોક્ટર ભગત સાહેબ હું વેલકમ કરું છું કે જે આજની વચ્ચે આપણી વચ્ચે ઉપસ્થિત છે. એમના જ્ઞાન અને અનુભવનો લાભ વિદ્યાર્થીને મળવાનો છે. નાકાઈ અને ફાલ્ગુની બેન એ સતત સાયન્સને જીવનને જાગૃત રાખે છે. તો અમારા યુનિવર્સિટીના કોમ્પ્યુટરના હેડ જે ફેસબુક અને યુટ્યુબમાં આખા પ્રોગ્રામને લાઈવ જાગૃત પંડ્યા, મારા ડિપાર્ટમેન્ટ ઓફ કોમર્સના સાથી મિલન શાહ, આ સૌના બિરદાવીએ. જાગૃત પંડ્યા એ અન્ય કાર્યક્રમોની વચ્ચે પણ સતત યુનિવર્સિટીની ફેસબુક યુટ્યુબ ચેનલને લાઈવ કરવા માટે એમનો સિંહ ફાળો છે. અત્યારે પણ પ્રોગ્રામ લાઈવ થઈ ગયો છે. જે વિદ્યાર્થીઓ અધ્યાપકો કદાચ આ કાર્યક્રમમાં જોઈન ના થઈ શકે તો પણ ફેસબુક અને યુટ્યુબ ઉપરથી આ કાર્યક્રમ નિહાળી શકે છે. યુનિવર્સિટીમાં અમૂલ્ય સ્મરણનું શું એ આવનારી પેઢી માટે અને આવનારા તેજસ્વી વિદ્યાર્થી માટે એક પાછું બનવાનું છે. 8,000 કરતા વધારે વિદ્યાર્થીઓ સમગ્ર ગુજરાતમાં આ પ્રોજેક્ટમાં જોડાયા છે. વિદ્યાર્થીઓ પાસે ચાર્જ કર્યો આખો નિઃશુલ્ક સેવા યજ્ઞ છે. બીજા અર્થમાં જ્ઞાન યજ્ઞ છે. નવરાત્રીના માં અંબાની શક્તિ ભક્તિનું અને જ્ઞાન શક્તિની આરાધનાનું કામ સતત છે. 4 મહિનાથી વધારે સમયથી ચાલી રહ્યું છે. કરનાર સૌ અધ્યાપક મિત્રો, મારા કોઓર્ડિનેટર સાથી ડોક્ટર રૂપેશ નાકર, ફાલ્ગુનીબેન પરમાર, સૌને અને વિદ્યાર્થીઓ જે જોડાયા છે એ સૌને હું ગુરુ યુનિવર્સિટીના SLET Ph.D એન્ટ્રન્સના કોઓર્ડિનેટર તરીકે એમનું સ્વાગત કરું છું. મને આનંદ છે કે અમારી ટીમ એક પણ દિવસ એવો ખાલી નથી કે જ્યાં વ્યાખ્યાન ના થતા હોય. છાત્ર ભવ એ અમારું સૂત્ર છે. બહુ જન સુખાય એ મંત્રને ધ્યાનમાં રાખીને અમે કરી રહ્યા છે. અને અમારી યુનિવર્સિટીનું સૂત્ર છે અમૃતમ તું વિદ્યા. એ વિદ્યા તું અમારા હૃદયમાં વસેલા અમૃતને બહાર લાવ અને ઓજ અને તેજસ્વી બનાવ અમને. અને એના દ્વારા ઉચ્ચ કારકિર્દી માટે અમે સૌ કટિબદ્ધ થઈએ. અમારો સહિયારો પ્રયાસ છે. આ એક જણનું કામ નથી. અનેક લોકોના પ્રયત્નોને પરિણામે આજે આપણે ખૂબ મંજિલ તરફ આગળ વધી રહ્યા છે. નેટ આ મહિનામાં ઓલરેડી ચાલી રહી છે. પીએચડી એન્ટ્રન્સ એક્ઝામ દરેક યુનિવર્સિટીની ચાલુ થવાની તૈયારીમાં છે. હમણાં ગયા 16મી તારીખે સૌરાષ્ટ્ર યુનિવર્સિટીની પીએચડીની એક્ઝામ થઈ. એમએસસી એન્ટ્રન્સ, પીએચડી અને ડિસેમ્બર જાન્યુઆરીમાં SLET એક્ઝામ આવવાની બાકી છે. એટલે આખો આ કાર્યક્રમ નિરંતર ચાલુ રહેવાનો છે. તો વિદ્યાર્થી મિત્રો, આ કાર્યક્રમમાં અમે તમારા માટે ખૂબ મહેનત કરીએ છીએ. અમારી ટીમ 14-15 કલાક કામ કરી રહી છે. તમે સૌ બિરદાવશો. મને લાગે છે આટલી ભૂમિકા પૂરતી છે. વંદે માતરમ, ભારત માતા કી જય. નાકર સર.
Thank you so much, Soni sir, for your very kind and inspiring word that you used, and you use each and every time to motivate us. And we all know that we have long way to go in the series of lectures in NET GSET preparation program organized by Sri Govindguru University. So, in continuation, we are in the 32nd lecture of NET GSET preparation series in life science. And today we have with us our esteemed speaker, Dr. Dehin H. Bhagat. And I would also like to welcome our coordinator Falguni Madam and two student representative Bharwad Vijay and Varya Jinil. Jinil will join us shortly in today's program. And I would like to welcome all the viewers who are watching us live on YouTube and Facebook through online mode. So today we have with us our esteemed speaker Dehin Bhagat sir. I would like to give short information biodata regarding Bhagat sir. Currently, he is working as an assistant professor in Srimad Raja Chandra Vidyapith at Dharampur, and he has experience of more than 5 years in teaching. And he did his PhD from Gujarat University on bacterial diversity of bacterial diversity of Tua and Unai thermal springs and their selected enzyme profiling. He submitted more than 20 sequence of nucleotide to NCBI, and he has good record of publication and has published more than 5 papers. He has also achieved Rajiv Gandhi Yuva Sanman award in 2020, and he is one of well-known member of Biotech Research Society. So we are very lucky to have you with us, sir. And as we all know that
Thank you so much.
Dehin sir is fond of using new technologies. So I think today he will use so many technologies to help all the students in understanding the concept of microbial genetics. Without delaying any time, I would like to invite Dehin sir to proceed for the lecture. Dehin sir, please.
Yes, thank you so much, sir. I'll share the PowerPoint presentation first.
Yes.
I hope, is it visible?
Yes, sir. Yeah.
Okay, fine. So without wasting much more time, I would like to thank each and every core member of this NET GSET MSc PhD entrance test program organized by SDGU Godhra. Particularly, I would like to thank VC sir for this initiative along with the registrar sir and the coordinator of the whole program for each and every subject. Along with that, Nakar sir and the Falguni madam, the way you people are working with your enthusiasm is really commendable, and this is the platform where I got the chance to thank my mentor, Dr. Prasanna madam. So thank you so much, madam, because I'm here because of her tribe. She always ready to help me and always be with me. So thank you so much, madam. So now actually this topic which I'm going to take today is microbial genetics. And as we all are aware that the genetics, it's quite lengthy topic, right? So this is my small try to make it in a concise form along with the animations and the technology, so you can it will be easy for you to understand, right? I'm using the bilanguage, right? Gujarati and English, so it will be easy for all the aspirant to make it for your NET and SLET examination, right? So Nakar are already introduced myself, so directly we'll proceed for microbial genetics lecture. So my voice and this display is fine, sir? Am I audible to all of you?
Yes, sir. It is completely fine. Please go ahead.
Great, great. So for each good work, we need something positive with us. So I will start my lecture with a few of the positive lines quoted by the Metsona Dilvayo. Basically, he's from Canada. It's a Canadian philosopher, and he shared one of the most inspiring quote with us. That is, the ordinary people pursue money, simple people pursue power, average people pursue fame, but the extraordinary people pursue ideas. This is really very motivating quote. So I thought that I want to put it here in this presentation for the aspirants, because students, you want to pursue for your bright future, you want to clear your NETs, late examination for PhD also. So every time, whatever is with you or not, but your ideas are always with you. So if you have a good idea, definitely your idea will change your life and you will be the extraordinary amongst the all other people. So this is my humble request to the all the aspirants, just take a photograph of it and keep it with you. Definitely this will help you for your upcoming examinations. It will always motivate you. And as you can see here, ETH cube, the E cube on the board, as you can see it over here, this E cube, this is the success mantra, right? Means what? It is having the 3E. So whatever the notepad you are using for your examination preparation, write down this meaning of E3 on the first page of it. Definitely this will be very much helpful for you. The first E, that is stands for exercise. Second is stands for enjoy. And the third E stands for explore. As you can see it over here, each kind of good work always starts with the exercise. If I'm here, if I'm taking your lecture right now, for that I have to prepare it last week. That is my exercise. But when I present it in a good way, where I enjoyed my work, right? Anymore I have to do. For that I need a proper exploration. So if you are experienced for your NET and GSLET exam, so yes, you have to recall these things. These are the very important positive vibes. Start hard working. That is your exercise. If you do a good job, if you do a good hard working, so along with that hard work, if you enjoy your work, it becomes very easy for you to understand. And after getting all the knowledge, try to explore more and more, apply your ideas, explore more and more reference material for your upcoming examination. Definitely, this is my guarantee, you will be on the top of the list. So these are the few things that I want to share with you regarding positivity. So with these all positivities, I will proceed for our lecture of the presentation of the today's topic. That is basically that comes under the, if you see the syllabus of NET examination, there are various topics in which one is inheritance biology, in which a small topic, that is the topic F, that is microbial genetics. So I'm going to discuss about this part only, right? So you already learned about the gene, allelic form, dominance, codominance, all the Mendelian principles, and almost all always linked with the plant and the animals, right? So now that is another world for you, and I will show you that world of microorganisms, right? I will explain this part specifically. For this lecture I will cover transformation, conjugation, interrupted making for you people, right? So because it is a long topic, so I will cover these three topics along with the problems, right? So problems means questions asked in NET and SLET examination. And if you cleared, if you are clear with this topic, definitely you will get 6 marks in your NET examination. Part B and part C, when you compare both the part, you will definitely get 6 marks if you are clear about these topics, right? So if you proceed for this, then as you can see it over in the coloring forms, these are the topics of our discussion. Our first and the most important topic is what and why. What is microbial genetics and why microbial genetics? I'm going to discuss about that part. Later on, we'll see the different types of processes, the gold standard procedures for DNA technology and this molecular biology. Apart from that, we'll see the implementation of that knowledge along with the problems, means question asked in your examination. I'll try to solve that, how to solve that questions in a limited time during the examination, and then I will give you the some reference material also, right? So this is the overview of my presentation, and now without wasting much more time, I will start with the main topic, that is the microbial genetics. I will start with the one beautiful story. Actually, that is not the beautiful, that is a drastic story. It's a dangerous story. But still, to understand the question why microbial genetics, absolutely we have to discuss about that part, right? So this is the brain map. This is called the brain map. As whatever you see here on this display, it is not a brain map. And the brain map requires so many things. The first thing is microbial genetics, and you can just simply explain this in It's ideal for studying the fundamental biological process. Up to now, you heard about many plant-related work on the maize, on the chickpeas, along with that Drosophila, right? So that is also called genetic study of genetic material, the study of the genes, study of the genomes. There are a lot of different types of definitions you will search. But here my focus is on microbial type, means study of the genetic material of bacteria or virus. So why it is interesting and what makes this very interesting for us, because Drosophila in maize you can see by your naked eye, but here in the case of bacteria virus you cannot see. Still, it is the ideal for studying all the fundamental processes in the biology. Now, let us start with the one important. This is the first point. That is the role of microbes for our survival. Just in a concise way, we all are aware that the microorganisms are our good friend. It always helps us. Just if we discuss production of yogurt, dahi banete, right? Then the second thing is nitrogen fixation, synthesis of vitamin B12 in our gut area. Now just imagine how means we have a lot of benefit from these microorganisms. Along with the certain very detrimental and dangerous things like disease and all, that is fine. But without microorganism, we cannot survive. And the study suggests there's no biota is a word where this organism means the body if in mice, suppose if we say gnobiotic mice, means mice or a hen without microorganism, is it possible for their survival? It is very difficult for the survival of that without the microorganism. So we all are aware that absolutely, if we want to live on this planet, we need a help of microorganism. Without them it's highly impossible, right? Along with this, if you focus on the Mendel's work, it's the no words for Mendel, because it requires a lot of patience, because microorganisms can divide into 20 minutes and you will get the growth within 24 to 48 hours. But Mendel, it's really heads off to Mendel, because he waited for the whole season. Working on plant is not an easy task, right? And the theory of heredity, absolutely, these two fundamental process, these are the roots for the development of the genetics. Along with the biochemistry addition, it means when you see at the biochemical level, all are made up of chemicals, right? All genes, if you know the chemical nature of gene, so everything is like up to just 4 macromolecules, and that emerges the field called molecular biology. And here we applied this molecular biology field on microorganism. That is why this is known as microbial genetics, right? And why it is very important? Because of the certain advantages of this. Suppose bacteria and virus, if you want to cultivate, it is very easy to cultivate, easy to isolate, easy to maintain, right? So there are certain benefits of this microorganism, and because of this, nowadays you will see the maximum technologies developed in the field of microbial genetics. And the most common is Escherichia coli, E. coli. We have a whole gene map nowadays. We have a lot of whole genome sequencing of many bacteria with us. So for the standard study, is a point of NET, SLET or any other entrance examination, you should know the basic genetics of these three: Escherichia coli, bacteriophage, and lambda. So I hope you people are clear about what is microbial genetics and important of it. Now why? Suppose if you see the story, if you see the past, beginning 19th and 20th century, beginning of the 20th century and the end of the 19th century is the era where you found a number of cases of tuberculosis all over the globe. And at that time, there were no cure for tuberculosis. Only the isolation is the way. So people tried to protect themselves by excluding the contact with the patient having tuberculosis. And all of you are aware, Mycobacterium tuberculosis is very deadliest bacteria which can cause TB in the human beings. But 1940 to 60, that is a time period where you will see, when you just go through that material, you will definitely understand that that is a period for the boom of antibiotics discovery. A lot of antibiotics were very well, mainly many like penicillin, streptomycin, right? That were very well studied during that time period, and that is the boon of antibiotics. People use that with without any manner, and that leads to multiple drug resistancy. So between the 1940 to 1991, people understood that now we eradicate TB from the world, because there were no cases at that time because of the use of antibiotics. But later on, if you see November 16, 1991, there is a headline in New York Times paper that a drug resistance TB results in 13 deaths in the New York. And then later on, through the, if you see the data, many cases of TB patients were recorded. So the scientific world is in confusion that what happened to this bacteria. It was sensitive, but right now it is resistant. That means something is there which makes this bacteria more powerful, more stronger in the terms of resistancy, and which encourages all the scientific community to specifically work on the microbial genetic side. During that time, we are aware about that the important thing is DNA. DNA plays very important role for this kind of inheritance changes, and this makes microbial genetics really very interesting subject for all of us. This is now, I hope you people are clear about what and why of the microbial genetics. So I will proceed for the one slide, that is the fundamental. Here in 1 or 2 line, I will explain the few of the fundamental with you. So how that TB bacteria becomes sensitive to resistance. We all are aware that is because of the DNA. In general term, that is a gene. So gene is basically from one bacteria to another bacteria. This is called gene transfer, and that is a very important scenario, right? So gene transfer, basically, if you study, there are two types of gene transfer we have to read. The first one is a vertical gene transfer. VGT means vertical gene transfer. So this is the one organism, just binary fission, you will get the two progeny, individual get the copy of DNA. It is VGT. But if you have one organism, this is the second organism, the genetic material of this transform from this bacteria to this bacteria. Suppose it is A, it is B. So transfer of this genetic material from A to B is termed as horizontal gene transfer. Clear? There are certain things we have to focus here. I hope you people are clear between the difference of VGT and HGT, right? Now, but here our main focus is on the horizontal gene transfer. This is the important for microbial genetics purpose. So it is always unidirectional. It never be a bidirectional, like as you see in the eukaryotes. In the eukaryotes, one chromosome, second chromosome, you can see the interaction, that is a bidirectional nature of the gene transfer, right? So that part is purely not happen here in the case of bacteria. Crossover. So suppose this is a chromosome, right? So this chromosome, if you see, is resistant. That means something is there. We make
Hello. Dehin sir, your voice is not coming. Dehin sir, your voice is not coming. I think we have some interruption, and soon Dehin sir will join us. Due to some technical faults, we are unable to join Dehin sir. Falguni madam, hello. મારો અવાજ આવે છે?
હા
ઓકે. તમે થોડીક વખત
હા
પેલું નેટની પરીક્ષા વિશેની થોડીક માહિતી આપી દઈએ આપણે. તમે જ આપી દો ને કઈ તારીખે છે ને બધું થોડુંક શેર કરીને 2 મિનિટ 3 મિનિટ જેવું.
ઓકે
I would request Falguni madam to present the newly declared date of CSIR net and some important features of the examination. Falguni madam, I would like to request her till Dehin sir joins us.
ઓકે સર ગુડ ઇવનિંગ એવરીવન Good evening, everyone. New examination date of CSIR has been announced recently, and examination of June 2020 will be conducted November 2020, and new dates are 19 November, 21st November, and 26th November. Dehin sir has joined us, so
Sorry
sir
for
will
interruption. There is an issue here with my actually I'm in a little bit of interior. That is why net connectivity is there.
no problem,
So
sir.
shall I continue with this?
Yes, sir.
Yes, but
Sir
I just
will
want to know
continue.
where. Yes, because at which part that's interrupted, I don't know. Can you help me for that?
Fundamental. The second part, unidirectional nature of gene transfer.
Okay, that is fine. So I will again discuss the fundamental part. Right now, I'm audible? Hello.
Sir.
Fine. So we were discussing about the few of the fundamental part for the microbial genetics. So first thing you should know about the gene transfer. Transfer means something passed from one to another. That is called the transfer. In the case of gene transfer, you will see two types. One is known as vertical gene transfer, another one is known as horizontal gene transfer. Right? Vertical means what? That is the bacteria. If you just read about the basic bacteriology, the binary fission is a mode of bacterial division. No, so this vertical gene transfer. This one you will get the progeny. Individual one get the DNA from this. That is the vertical gene transfer where you have two bacteria. So DNA or any genetic material transfer from one to another. That is the horizontal gene transfer, and that should be unidirectional. So this is a very important point. Note it down that in bacteria, the gene transfer is always unidirectional. In the case of eukaryotes, that is bidirectional. And if you talk about the crossovers, right? Later on in the theory of conjugation, I will explain how these crossovers happen in recipient bacteria, right? So just write it down the main important points. So this will be helpful for you. The third point is all about bacteriophage. Bacteriophage means what? Bacteria which can infect the bacterial cell, virus which can infect the bacterial cell, right? Animal virus can infect animal cell, plant virus will infect plant cell, and bacterial virus which can infect bacterial cell, that is known as bacteriophage. So what happened in the case of bacteria? Certain bacteria like T4 phage, when infects the bacterial cell, particularly E cell, it injects the DNA. This DNA takes the control of the mechanism or a machinery of that specific host cell and degrade the DNA or degrade the chromosome of host cell and produce its own proteins, right? For new virion particle, and at the end, one infected cell will produce 300 viral particles, and ultimately that leads to the lysis of that host cell. જો ગુજરાતીમાં સિમ્પલ શબ્દોમાં તમને સમજાવ તો ઇશેરિશિયા કોલાઈ ઈ કોલાઈનો સેલ, રાઈટ? એમને જો એક બેક્ટેરિયોફાજ એક T4 ફાજ એને ઇન્ફેક્ટ કરે તો એમાંથી 300 નવા વાયરસ પાર્ટિકલ બનતા હોય છે, રાઈટ? 300. And basically the DNA of that phage particle will control the whole mechanism of that host cell and produce its own protein, and ultimately that degrades the chromosome of bacteria and releases from the bacteria, release the newly synthesized part, and this is known as lytic cycle. And the second one is known as lysogenic. If we see the lysogenic, the infection mechanism, just like it's the same, when this viral, like it is the lambda phage. If you want to study the lysogenic cycle, that is the lambda phage. If you study the lytic cycle, that is the T4 phage. This lambda phage DNA is integrated with the chromosome of the host cell, right? Integration is important here. I'll use the different color so it will be easy for you to understand, right? So this green part shows the integration of viral DNA with the chromosomal DNA of Escherichia coli, and it persists for a long time. That sometimes it is known as prophage, sometimes it is known as prophage. Clear? So there are two things to remember. One is the lytic cycle in which cell lies, and the lysogenic cycle where the part of that viral particle remains stable as a part of the bacterial chromosome for many generations. That is called the lysogenic cycle. Why these are different? Why these are very important for microbial genetics purpose? Because during this integrations, many bacterial genes plays very important role in the gene transfer here, right? So we will discuss that part later on. Now, another important topic is bacterial mutation detection. How you detect mutation, and this is the very important fundamental for the interrupted gene medicine. Suppose, in short, if I'll explain here with the short discussion, just I change the color of the pen. Okay, fine. So, source of energy. If I'm talking about bacteria, right? Bacteria require some kind of sugars for their source of energy. For example, lactose sugar. If you talk about the essential metabolites like tryptophan, arginine, if you see the resistance for drug against streptomycin. So whatever the bacteria is, if you know their utilization pattern, you can easily check the detection. How? I'll explain it over here. Suppose you have a one bacteria which is able to produce, which is able to use lactose and galactose as a source of energy. Okay, your bacteria is able to use this. So this is known as lac plus and gal plus. લેક્ટોસ યુટિલાઇઝ કરે છે, ગેલેક્ટોસ યુટિલાઇઝ કરે છે. Fine. So, but due to the certain mutation, just try to recall one practical of UV mediated mutation, right? So if UV mutation is there and that disrupt the gene of the lactase beta galactosidase enzyme, which can use for the degradation of this lactase, it gets disrupt, then that organism is not able to produce lactose. So simply just try to grow that bacteria on the plate containing lactose as a sole carbon. If your organism is not able to grow, that means there is a mutation in the lag gene. Same way for essential metabolites, try to grow that bacteria in the minimal agar plate where no tryptophan or arginine is present. If it requires that essential metabolites, means it should have some kind of genes in its chromosome, right? And if you see the drug, suppose the organism is not having resistance against streptomycin, means it's a sensitive one, but due to the mutation, some kind of mutation, it becomes resistance. So you can check it by growing this kind of culture on minimal agar plate with streptomycin. એટલે પ્લેટ છે એની અંદર મિનિમલ મીડિયમ છે અને એની અંદર આપણે એડ કર્યું છે સ્ટ્રેપ્ટોમાઇસિન. જો સેન્સિટિવ હશે તો ગ્રો નહીં થાય, રેઝિસ્ટન્સ હશે તો ગ્રો થશે. એનો મતલબ શું થશે કે રેઝિસ્ટન્સી એને મળી છે. The same phenomena we will use for the interrupted gene MAGPIN, right? I will explain that part in depth, so this will be easy for you to understand. And the fifth important point is plasmid. There are two types of plasmid. Very important part. You have to focus here. One is R plasmid, one is F plasmid. Resistance plasmid and fertility plasmid. Here our F plasmid is important. And the plasmid is the extra chromosomal part of the DNA. Means if you see the bacterial cell, the important thing is the chromosome. But along with the chromosome, some bacteria is having the extra chromosomal piece of the DNA, which is having very important genes, is known as the plasmid, right? If it is independent there, it is known as plasmid, but certain plasmids is having ability to integrate with this chromosome. આ ક્રોમોઝોમ સાથે ઇન્ટિગ્રેટ થઈ શકે છે અને એવા પ્લાઝમિડ માટે એપિઝોમ વર્ડ યુઝ થાય છે, that is એપિઝોમ, right? Right, so that is episome. So I hope you people are clear about these things. I just want a reply from you so I can easily make it out.
Yes, sir. Please continue.
Okay, great, because I want to check sound at each slide because of the last experience. Fine. So after the discussion on the fundamental, we are too much clear about the gene transfer part, unidirectional process, two crossovers, bacterial lytic and the lysogenic cycle, fundamental of plasmids, and the detection of bacterial mutation. The same phenomena here we will use it to check the gene transfer. How? That we'll discuss now. Okay. So basically, there are main mechanisms. All these mechanisms are known as parasexual mechanism. Why? Because here you will not see male and female like, okay, still many papers if you go for the jam paper or NEET paper, you will find female E. coli or male E. coli word, right? But most commonly in the NET you will not find this kind of terminology, because in bacteria there isn't any kind of male or female bacteria, right? It is the F plus and the F minus bacteria. So we'll discuss that part later on, but it is not at all sexual mechanism of gene transfer. That is why it is known as parasexual, right? So there are three main types of mechanism. Just try to focus here. The first one is known as transformation. It is a very important procedure. Transformation, where a naked DNA, only DNA is present in the environment, and that DNA uptake by your cell directly. So this uptake of free DNA, and this mechanism is known as transformation. Very simple, you have to recall it. When we talk about the free DNA and its uptake from the environment, this mechanism in a simple term known as transformation. Now here only one bacterium is there. No gene transfer between two different organisms, two different bacteria. It is just the naked DNA. The word for the free DNA is naked DNA. Okay. So one mechanism is transformation. As you can see here, it's a naked DNA means free DNA. A DNA not transfer. So that is why mechanism of gene transfer, genetic material transfer, right? Now, if we focus on the second one, that is the conjugation. Here you can, there are two types of cell. This is the first cell, this is the second cell. You will see there is a bridge-like structure between these two. Means what? For the conjugation, the physical contact between two bacterium is necessary. So this bacterium joint with each other. It is a very layman language terminology, but for better understanding, they join through some kind of bridge or canal, and from that canal, the genetic material will be transferred. So this transfer from this side only, unidirectional only. And this mechanism is known as conjugation. If you see the third one, that is a transduction. And transduction means what? I already mentioned the lytic cycle. This is the lytic cycle, kind of. So new progeny releases after the lysis of the host cell, and this will infect another cell. But here, if it is a pure lytic, it will not transfer bacterial DNA because that stretch of DNA is never integrated with the chromosome of bacteria. So lysogeny, at the end of the lysogenic cycle, when that part of DNA cured from the chromosomal part, so this is the viral DNA. Along with the viral DNA, sometimes it gets the fragment of bacterial chromosome. So this bacterial chromosome, part of the bacterial chromosome, transfer from one to another, and that procedure is known as transduction. So the most commonly, if it is a naked DNA, that is a transformation. There is a physical contact, that is a conjugation, and through virus, that is known as transduction, right? So there are two important criteria to distinguish between all three types of mechanism, right? Now ask this question to yourself first. Does this process require cell contact? If we say for the transformation, ask this question for transformation. Does this process require cell contact? And we can say no. Here, yes. And for here, no. Now the second question, is this process, if you see this process, so this is sensitive to DNase and enzyme. DNase is an enzyme that can degrade the DNA, right? DNA molecule ને એ શું કરી નાખે, break કરી નાખતું હોય છે, right? So is this process is sensitive to DNase? અહીંયા તો naked DNA છે, so definitely this is sensitive. So for the second question, this should be yes. For this here, your DNA is covered here. These are particular these. Bacterial cell wall and plasma membrane protects this DNA. So is it sensitive to DNase? I can say no. Here in this case, DNA is covered by the protein coat of the virus. So here also no. And so these two criteria can be tested experimentally, and I will discuss this experiment with you. So I hope you people are clear about these two questions and all these three types of parasexual processes. Now let's move towards the theoretical part. So how you check whether the process is transformation, conjugation, or transduction, right? So the whole scenario starts with one experiment. Suppose you have a culture. Here I will explain one by one. So this will be easy for you to understand, right? So suppose I have a culture. As you can see it over here, in this culture tube, I have a culture. Okay? Culture along with DNase enzyme. So let us check. In this tube, I already added DNase enzyme. So if the naked DNA is present in that bacterial culture, so this particular DNA degraded by your DNase enzyme. Now no DNA, then no gene transfer, right? It disproves that the procedure is transformation. Suppose, I'll explain it in a simple language. You have a tube, right? And when you check the gene transfer, okay, this is a tube. After a certain time, you plated this culture on the nutrient agar plate or any other plate, and you will see yes, this particular organism will get certain new characteristics. So that mechanism is transformation. But how you prove that this is transformation? It may be transduction or conjugation. So for that, just do one thing. Take your bacterial culture, add DNase enzyme, wait for a few minutes, check for gene transfer. If you will find no gene transfer, and that suggests the mechanism is transformation, because the DNA which is in the naked form is not available. Why? Because of the activity of DNase enzyme, right? And why this type of things not happen in the case of conjugation and the transduction? Because as I mentioned earlier, protein code of bacteriophage and the cell wall and cell membrane of bacteria will protect DNA against this DNase enzyme. I hope you people are clear, right? So this is called transformation. Once again, I will explain. If you have a tube in which you have your bacterial culture along with the DNA, incubate it for 24 hours. Next day, you will see some new characteristics that suggests there's the transformation procedure. But you have to prove that whether it is a transformation or any other cells. So select the same culture and add DNase enzyme and wait for the 24 hours and check for the gene transfer. Next day, you will not get gene transfer. That suggests that the mechanism is only and only transformation, right? So how you can check whether the procedure is transformation, conjugation, or transduction? Transformation we check. Now see, this experiment is a very fundamental experiment in microbial genetics. That is the YouTube experiment, where you have a U-shaped tube, where this tube, you have two types of culture. One is A minus, B plus bacteria, means it is having two type of characteristics. A minus means, suppose A is your lactose and B is your streptomycin related. So A minus means this culture is not able to utilize lactose, but they resistance against streptomycin. The same thing here, lactose utilize, yes, streptomycin sensitive. So you have to fill this YouTube with these two culture, and both the cultures are separated through sintered glass filter. Okay, so this filter is having the pore size which can allow only DNA and virus, but not the bacteria. So જે બેક્ટેરિયા છે આ ફિલ્ટરમાંથી પાસ થઈ શકતા નથી. પાસ તો થઈ શકે ખાલી DNA and virus. Okay, now what you have to do is, once again, આ જે કલ્ચર છે એને પ્લેટ પર પ્લેટિંગ કરીને ચેક કરો. જો A પોઝિટિવ છે, B નેગેટિવ છે, એની જગ્યાએ તમને A પોઝિટિવ કે B પોઝિટિવ મળે, right? If you will get A positive and B positive, means something genetic transfer happen over here. Now you have to check whether it is transformation, transduction, or conjugation. How you check? Because your earlier strain is A plus B minus, and now it is A plus B plus. So something transfer here. So for that check, take this part, add same thing, repeat this, add DNase in it. Next day, no gene transfer, the process is transformation. But gene transfer, yes, your answer is yes. Then what to do? Now there may be a chances of conjugation or transduction, right? You have two options. If gene transfer didn't happen, then that is a transformation. But here in this case, gene transfer occurs. Now you will have to check whether it is belongs to conjugation or transduction. So for that, if it is a yes, then what will be my answer? My answer will be transduction. Why my answer will be transduction? Because here I know that for conjugation I need a physical contact, just like this. As you can see, two bacteria physically attached with each other. So already there is a sintered glass filter, so there is no physical contact, so it rejects the probability of conjugation. So ultimately it proves that the mechanism behind this genetic transfer is not a transformation or not a conjugation, and only transduction. Why not transformation? Because we already added DNase enzyme and still genetic transfer occurs. Why not conjugation? Because the sintered glass filter is here, so no physical contact, so no chances of conjugation. So only the mechanism is transduction. So this transfer of DNA happen here because of the viral transfer. That is the transduction. right? So if we see the E. coli, so in E. coli, transformation is not possible, because in all the bacteria, it is not possible all three types of genetic transfer you will see in all the kind of bacteria. No, that is not at all possible. Certain bacteria is their own mechanism. Suppose if I'm talking about E. coli, so you cannot see transformation in E. coli. Why? Because for the transformation, certain proteins are required, and that proteins are absent in E. coli. So you will never see a transformation in E. coli. If you want to do in lab, you need some physical and chemical methodology. મારી મારીને મિયા બનાવવાની કહેવત છે ને, એ એપ્લાય થાય. મીન્સ E. coli naturally not possible for transformation. Yes, conjugation you can see in E. coli, right? Transduction you can see in all kind of organisms. So for all the bacteria you can see transduction, but transformation very few, right? So why E. coli is a model organism for this? Because it shows conjugation, and that is a very important mechanism to understand this genetic transfer. So we choose E. coli, right? Now all of you are clear, sir? Am I audible?
Yes, sir. Yes, sir. No issue.
Great. Right, because I need some interaction at the time of discussion. Otherwise, it's a unidirectional only. For the presentation, I need some bidirectional. So it doesn't matter. So it's working fine. So up to now, we are clear about the type of genetic transfer, all the three types of parasexual cycle. The first one, transformation, naked DNA. Second one is conjugation, required the physical contact. And the third one, that is transduction, requires the viral particle, right? And we already checked through the experimental procedure, which is known as YouTube experiment. I hope you people are clear. Many times, students, you will get questions from this part, YouTube experiment part, right? They will check, they will ask the question, and you have to guess which kind of procedure occurs in this particular phenomena. They will explain the whole procedure, and you have to guess whether it is a transformation, transduction, or likewise that. Okay, so this is about the introduction part of that. Now we'll focus on the original mechanism, right? Part 2. This is related with the part 2. So in part 2, you will get part B of NET examination. You will get 1 or 2 questions from this, at least one each year. For JEE NET, people ask for 12th standard also sometimes. But for our MSc and PhD exam, this is the most important topic, particularly if you want to pursue for PhD in microbiology. Yes, at least 4 or 5 questions we will see from this particular microbial genetics unit, right? Now we'll see the transformation procedure in a very concise way. So the transformation, when we discuss about the transformation, we always remember the legendary Frederick Griffith, the scientist who did a fantastic job in the field of molecular genetics, and he's the first who gave this kind of idea, because in the beginning of our presentation we discussed about the idea, right? So what happened in the gene transfer, the first idea was given by this Frederick Griffith. He never coined this terminology known as transformation. So basically he was working with the Streptococcus pneumoniae, that is the bacteria which will cause pneumonia, right? In general it is known as pneumococci. So he worked on the pneumococci, and during the microbiological work he basically observed the 2 different phenotypes, right? He observed, because at that time we don't have a genotype and all type of fundamentals. So basically everything is based on the phenotypic characteristics, right? And he found this genetic variability particularly in the phenotypic characteristics, and his main focus is on the EPS, that is the exopolysaccharide. Nowadays a generalized term is known as capsule. Okay, so when he cultivated this pneumococci in the lab, he observed that there is a variety of phenotypic characters in pneumococci, right? How? So basically he cultivated these organisms on blood agar plate, right? On blood agar medium. I hope you people heard this name of this medium, blood agar plate. So when he cultivate pneumococci on blood agar, he found 2 different types of morphology. One type of morphology, you can see it over, quite big colony in the terms of size, that is big, and one small size colony, right? So he checked that whether there's this exopolysaccharide material, this capsular material is present or absent. The first point he observed is the presence and the option. And the second one he observed about the molecular composition of that particular capsule. Clear? So what is Griffith's main focus? Griffith's main focus was on capsular material is present or absent, and the second one is its molecular composition. And based on that, he termed huge capsulated colony as a type S colony, and the small without capsule, that colony designated as type R. So he worked with type S colony and type R colony. And these 3 and 2 are the see, each antigenic each capsule is having its own antigenic properties. Based on the antigenic properties, there are varieties of types in pneumococci. So basically he works with type 3S pneumococci and type 2R pneumococci, right? So there are type 1, type 2, type 3, many types of antigen, means that here type mainly focus on the molecular composition. Based on the molecular composition, you can classify that into type 1, 2, 3, 4, and 5. Just you have to recall that type 3S and the type 2R, right? These two were very well observed by Griffith. Now what he did, the experiment, I will discuss this experiment. This is known as the right? What he did at that time, so first he isolate and maintain the type 3S pneumococci in the lab. And what he did at that time, he inject that type 3S into the live mouse. And wait, what happened to that mouse? The ultimate thing is, he found that mouse is having pneumonia, and that leads to the death of that mouse. And he recovered type 3S from the carcass of that mouse. Carcass એટલે શું dead body એમ કહી શકાય, right? જે mice જે મરી ગયું છે એની dead body માટે word use થાય છે carcass, right? So he recovered type 3S from that dead mice. Side by side, what he did, he boiled that culture of type 3S. He killed type 3S and inject in the live mouse. Once again, the observation is very important thing over here. And what he found that the mouse live. Why? Because that pathogenic culture of S. Yes, pathogenic means what? He also found that pneumococci with the capsule is pathogenic, but pneumococci without capsule is not pathogenic. Pathogenic એટલે એવા bacteria જે disease cause કરે છે, right? So when he killed type 3S using the heat, that culture is not able to cause pneumonia in the mice. Mice survive. Third set he took R. As I mentioned, R is non-pathogenic. When he inject this strain in the live mouse, it becomes healthy. No pneumonia in that mouse. But scientists are always scientists, you know. So what he did now, the fourth part, he mixed this live type 2R with heat killed type 3S. He mixed these two in a one test tube and inject that mixture in live mouse. Results were outstanding. Mice died because of pneumonia, and he recovered type 3S. Right? How it is possible? Because type 2R, how it is converted into type 3S? Because type 3S is already in the killed position. Means what he realized, he concluded that there is something which transferred from type 3S, heat kill type 3S, to type 2R into type 3S. Suppose if it is a mutation, then that type 2R is converted into type 2S. It is very clear and very easy to. If you have a type 2R, if there is a mutation only, then it is type 2S. But here it is not at all mutation, no chances of mutation, because type 2R is converted into type 3S. S. So mainly type depends on the molecular composition, and molecular composition always based on these genotypic characteristics. So it is proved that this is not the mutation, but something is transferred from heat kill type 3S to type 2R, and which gives a superpower to this type 2R. And so because of that, that is converted into type 3S. And type 3S is having the capsule. So it's a virulent, and ultimately that leads to the death of that mice. And this is known as the transforming principle. This term was coined by Griffith. He concluded this transfer as a transforming principle. Everything is a matter of 1920 to 2030, but in 1931, Richard Shea and Martin Dawson, they coined this term as a transformation. Means this transforming principle, the mechanism is known as transformation. But still, up to 1931, people are not aware about permanent inherited change. Here, scientists found that this transforming principle leads to the permanent inherited changes. Means type 2R is converted into type 3S, and that is a permanent one. So this permanent inherited change that was proved by Oswald Aubrey MacLeod and McCart in 1944 that this permanent inherited change is because of the DNA, and that is why now we are aware about this thing. The transforming thing is your DNA, means gene, right? So Griffith demonstrated very well, and after performing this, you people are now clear what is transformation, what is transforming principle, what is the proper explanation of this, because this part, the written part in the red color, is very important. This distinguishes normal mutations from this transformation procedures, right? It is due to the gene transfer, not because of the mutation, because type plays very important role. Molecular composition, phenotypic characteristics plays very important role. I hope this Griffith experiment is clear for all of you, right? Clear? Yes, sir.
Yeah, sir, please continue.
Okay, great. Now this is about the Staphylo, just a pneumococci only. Now, if you just refer the past few papers, you will see certain other bacteria and the transformation related with them. So here I included one is Streptococcus pneumoniae. Another important bacteria is Bacillus subtilis. Definitely you will find many questions from Bacillus subtilis also. So in Bacillus subtilis, it can take up DNA from any source, right? So no restriction here. But if you see the Haemophilus influenza and Neisseria gonorrhea, so that organism, both are gram-negative organism, and they take up their own DNA or a DNA from the closely related species. And the uptake capacity is just 11 base pair in Haemophilus and 10 in Neisseria, right? So what makes this bacteria competent? The word is competent. Competent means what? There are certain kind of protein called the com proteins. So the organism which is having a capacity to catch up or to take up DNA from the environment is having specialized gene for com proteins. These com proteins. એટલે સિમ્પલ વેમાં કહો તો જે બેક્ટેરિયા પાસે કોમ પ્રોટીન હોય એની અંદર જ તમને ટ્રાન્સફોર્મેશન જોવા મળે. જે સેલ રેડી થયા હોય એના માટે એને કહેવાય કમ્પિટન્ટ સેલ. Not all cells are competent, mind it well. Only cells who is having this mechanism, who is having this protein, are able to uptake the DNA from the environment. Now another question, at what time this kind of bacteria will accept the DNA? So that is mainly the late phase of their growth cycle. So at the end of the growth cycle, late phase of the growth cycle, where cell density is very high, number of cells are high, but the division stops. At this particular time, Bacillus subtilis specifically accept the DNA from the environment for the survival purpose, right? And at that time, the important protein is pheromones. So this higher concentration of pheromones was found at the time of late phase, and these pheromones induce the genes for the COM protein production. એટલે શું થશે? At the late phase, you will see the higher concentration of pheromones in bacillus subtilis, which motivates these genes for the production of COM protein. એટલે આ genes એ COM protein બનાવશે. આ COM protein ક્યાં જશે? એના સેલ મેમ્બ્રેનમાં જશે અને ત્યાં એ બાઇન્ડ થશે અને ત્યાંથી ડીએનએ ને અંદર સેલની અંદર એન્ટ્રી આપશે. Right? So I will discuss one simple procedure of Bacillus subtilis, the mechanism of transformation in Bacillus subtilis. Now just focus. I use this term, competent bacterium. Means what? It itself indicates it is having certain proteins for the acceptance of the DNA from the atmosphere. So this is the chromosome which is having the A negative gene, right? So this is your Bacillus subtilis with this A negative gene and certain proteins for acceptance of DNA from the atmosphere. Now what happened? It is having the DNA receptor in the atmosphere or in the environment. This is the gene which is having the A plus gene. It's a naked DNA. Mind it well, it's a naked DNA, or else we can say that it's a free DNA. So what happened next? This free DNA binds with that particular DNA receptor. Now let's zoom this protein. When you zoom it, now you just focus here on this second figure. I hope you people can see my cursor, right? So this DNA is having A plus gene. This is the extracellular environment where gene is here. So first, two proteins, COM-G protein and COM-EA protein. These two important proteins which properly binds with the extracellular DNA and align it for the proper entry inside the cell, and which are present, both these proteins are present in the cell wall only. What next? We all are aware that naked DNA is double-stranded DNA. So you will see the deoxyribonuclease enzyme in between the space between the cell wall and the cytoplasmic membrane, paraplasmic space. So here in this space, you can see the deoxyribonuclease enzyme, which can degrade the single strand of that particular double-stranded DNA. So only one strand is able to get entry inside the cell. Other one is degraded by deoxyribose nuclease. Now you can see two more proteins here, COM-FA and COM-EC. These are the translocase activity, means these two are able to transfer it from outer to inner. So it stabilizes the DNA, and along with that, single-stranded binding protein. Just try to recall the theory of replication. So during the replication, single-strand stabilization is mainly due to the presence of single-stranded binding protein. So these are the protection mechanism for single strand which enters from outer to inner side of the cell. And another protein are RAC-A protein. RAC-A protein are useful for the integration of the single strand in bacterial chromosome. Just go ahead. So that is the second one. Now, with the help of RAC protein and other proteins, you just see in the here, right? A minus is there. So your DNA single strand replace the single strand of this chromosome and integrates over here. This integration requires a lot of proteins, right? So now you can see the single chromosome is having two allelic form, A minus and A plus. Both the forms are there in this form, right? That is why it is known as heteroduplex. And this the single chain which is detached from the chromosome is then degraded, right? So this is how the naked DNA gets entry inside the Bacillus subtilis and integrated with the bacterial chromosome. If you see it over here, right? So this is the heterozygous. Now just focus vertical gene transfer, one cell and the second cell. Now both the cell, if this divides into two, each cell will become homoduplex instead of heteroduplex, because A minus ની સામે નવી સ્ટ્રેન્ડ બનશે, A plus ની સામે નવી સ્ટ્રેન્ડ બનશે, right? So A plus ની સામે એના complementary, A minus ની સામે એની complementary. So one is having the two A plus, one is having A minus. So it becomes homo. Clear? Homo duplex. And if you focus here just, so DNA molecules of this what is the quantity of this DNA? Because if the DNA is too huge molecule, your cell will not accept the huge molecule. It just accept the 0.2 to 0.5% of the complete chromosome. Suppose 100 base pair, so 0.2 to 0.5% of that 100 were accepted only, right? So this is the very important question. As per part B question, yes, how much amount or a percent of naked DNA will be accepted by your cell? That is 0.2 to 0.5%. And I hope you people are now clear, because according to your basic syllabus of microbiology, you have up to pneumococci only. But for NET, SLET, or at least MSc entrance or PhD entrance, you require this mechanism, right? So I hope you people are clear about the whole transformation procedure in Bacillus subtilis. I request all participants to take a snapshot of it, so it will be very much helpful for you. Now we'll proceed for another important topic, that is conjugation. Yes, sir. Am I audible?
Yes, yes, completely. Please go ahead.
Yeah, so right. So we have 519. Okay. Time plays a very critical role over here. So now I will discuss with you about another important mechanism of gene transfer, that is known as conjugation. Now you people are clear that conjugation is a very important technique for the gene transfer. Particularly we talk about E. coli. So E. coli does not occur, means in E. coli transformation does not occur, right? So in 1946, these are the another two scientists whose role in the development of microbial genetics is really commendable, right? So Joshua Lederberg and the Tatum, these two scientists discovered that the E. coli cells transferred genes by conjugation. Now you people are clear about the mechanism, what happened during the conjugation. I already mentioned, for the conjugation purpose, the very important thing is the physical contact. Just hold on, my pen is not working, I don't know why. Just hold on, the graphic tablet is not working. Right, now it is working. So for the conjugation, what we require the physical contact. As you can see here in this figure, this is the electron micrograph. There are two cells. You can see the F plus word over here. You can see the HFR H word here and F minus 1. I will explain each terminology with ease, right? But the important thing is this highlighted part. You can see the physical contact between these two cells for the gene transfer, right? So from this is the direction of gene transfer. F negative will get DNA from this HFR H, right? So this conjugation methodology has proven to be the most important method. What? Genetic mapping. Mapping is what? We'll discuss it later on, but it is a very important tool in the genetic research, right? So what happened during this conjugation procedure? Let us see one by one. Okay. So here the important thing is F factor. If we talk about the E. coli, the first and the most important thing is F factor. F factor is nothing but the kind of a plasmid. Mind it well, it is a plasmid only. Plasmid which is having certain types of gene which makes this E. coli cell donor. Means the cell which is having this F factor can donate this DNA. Let's see. Suppose this is the E. coli cell, and we all are aware that it is having the chromosomal DNA. So the red color is your chromosomal DNA, right? But it is not having any F factor, or which is not having any gene, right? So I'll just erase it now. Just focus over here. So in E. coli cell, where you will see only the chromosomal DNA, right? no other kind of extra chromosomal DNA or plasmid in it. So that is known as F cell. F minus cell. Minus means what? It lacks the F factor. Suppose this is another cell, chromosome in it along with the piece of extra chromosomal DNA, that is called F factor. So this particular black circle cells of F. And these kind of cells are known as F cells. Right? So you have to be clear which kind of cell is known as F minus and so cell which is not having any F factor is known as F minus factor in it. The chromosome inherited type and they are settled in it from the chromosome. And the third type of cells we will see here in this case, HFR cell. High frequency recombinant cell means these are the cell in which your F factor is integrated with your bacterial chromosome. As you can see it over here, it is independent. Here, in the case of HFR, you can see your F factor is integrated with the bacterial chromosome, and that is why this type of cells are known as HFR, where it is independent. This is known as F plus, and this is known as F minus. So I hope you people are clear about the difference between F minus, F plus, and HFR cell. And this HFR, as you can see, it is a part of the bacterial chromosome now, and that is why it is very important for genetic mapping. Now we'll see, because it is a covalently bind, very firmly bind to the chromosome. So detachment of this from this is also a very difficult task. So now one word is known as donor cells. Donor cell means what? I already told you earlier. Cell which is having the F factor is known as donor cell. ડોનર કોણ હોય જેની પાસે કંઈ હોય આપવા માટે એને ડોનર કહેવાય. સપોઝ આઈ વોન્ટ ટુ ડોનેટ મની બટ મારી પાસે પણ હોવા જોઈએ ને, રાઈટ? સો ડોનર સેલ મીન્સ એટલે તમે જો ટેકનિકલી પણ જો તમે વિચારો તો ડોનર સેલ contains something which can be donated by that particular cell. So the F plus became donor cell or HFR became donor cell because both this type of cells is having this F factor in it. So the cell containing F factor known as donor cell. Now, what is the role of this F factor? So F factor is having certain kind of genes which can produce F pili, and this F pili is required for physical contact. Okay, and it is F factor. Why it is known as F factor? Because it helps for parasexual mechanism called conjugation. That is why it is known as fertility factor, and this is the size of that F factor, right? It is a small circular molecule. I already explained over here, right? Now see, here orange color, this one is your recipient cell which is not having the extra chromosomal DNA. The blue one is your A plus cell which is having the F factor. Now for this contact, F pillars plays very important role. So these are the F pillars. So F plus cell will produce this kind of F pillars which binds to the nearer recipient cell, right? So this helps for the contact. એકબીજાની નજીક લાવવામાં કામ કરે છે, right? પહેલા શું હતું? Earlier version, right? I'll explain this. In the past, what is the observation of the people? So scientists concluded that the transfer of this F factor from this is because of this F pillars. But due to the advancement in the techniques, nowadays we know that the role of these F pillars is just like a rope to pull two cells together. એમને નજીક લાવવા કામ કરે છે. But the transfer of F factor is basically through one channel called conjugation channel, right? So there is a conjugation channel form. Here is a blue arrow. It is the conjugation channel. So from this channel, this F factor will pass from this cell to this cell. and the both cell become F plus at the end of conjugation. જો conjugation પતી જાય એટલે આની પાસે પણ એક copy રહેશે અને આની પાસે પણ એક copy રહેશે. So how this transfer, right? Because it is a double stranded. So only one strand is able to pass from this. One strand remains here only. So this will receive one strand. It is having already one strand. So due to the replication, it becomes double stranded and both becomes F plus. Clear about these things? So this is how conjugation occurs. Now if we focus on the F factor, these F factors are of two types. બરાબર છે, બે પ્રકારના F factor હોય છે. One is known as the autonomous state. એક state છે that is known as autonomous state. Autonomous state means what? Just like you can see it here in the case of F plus. This is the autonomous state. બધા કરતા અલગ, એની જાતે replicate થશે independently, right? That is the autonomous state. And the second state is integrated state. That is the, as per my explanation of HFR. So it is integrated with the bacterial chromosome, right? So there are two states. આના પણ question આવશે નેટમાં about the state, right? So one is autonomous state, another one is integrated. If you talk about the F plus, that is the autonomous state. If you talk about HFR, that is the integrated state, right? So the next one is about the F factor. So the integration, this is the integration, how it binds with this chromosome. So this integration of F factor is mainly mediated by the short DNA sequences, right? This is known as insertion sequence. If you focus here, it is known as IS. Insertion sequence, specialized types of sequence which is present on HFR also and which is present on the bacterial chromosome at the multiple sites. So this F factor can integrate with bacterial chromosome at multiple sites. So this multiple site integration can be possible here, which makes a different type of HFRs, HFR1, HFR2, HFR3, based on that integration, right? Now, so this kind of physical contact is possible between F plus and F minus, because F plus will give that part of DNA to F minus. This is known as F plus into F minus mating, and HFR can donate. So HFR into F minus, right? So there's mating. There are two types of matings occurs here. F minus ને આપશે, F plus to F minus, and HFR to F minus, right? And for these are the site. If you talk about, this is your F factor. So F factor is having a specific site. Suppose this site is ORI-T site. So this site is required for the replication purpose. So DNA replication can be initiated at this site, and this site is known as ORI-T. Clear? So this is a site of replication initiation, right? So this is the ORI-T. One site is known as ORI-V. ORI stands for origin. Origin means કશું શરૂઆત થવી. ORI-T is for DNA replication initiation site. ORI-V, that is the primary origin of replication during the cell fission, right? Fission and right? This is the site fission. This is the site at the time of cell division. At the time of cell division, initiation of replication starts from ORI-T site. At the site of fission, this starts with the site V. And ORI-S, then we have one more question. What is the importance of this S site? So secondary origin site works when this ORI-V is absent or non-functional. So in the absence of this, because certain mutations or something leads to the deletion of that ORI-V site from the F factor, at that time ORI-S, that is the backup procedure for the bacteria, right? Now we'll see F plus F minus mating one by one. So how it starts, just focus, you have a two types of cells over here, right? I'll see the time also, because we have only, okay, it's 5:32, we have half an hour more. So if you focus here, just title is as a 1. So this is your F plus donor cell, this is your F minus recipient cell. So how these two bacteria mate with each other, that is the conjugation procedure. Okay, so it is having F factor, it is not having F factor, right? So this is the first step where the F pilus pulls these two bacteria together. એકબીજાને અહીંયા નજીક લાવવાનું કામ કરે છે. Now the next step is what? After that, there is a formation of conjugation channel. There is a bridge between F plus and F minus. Okay. After this bridge, you can see it over here. F factor is having a ORI-T site. So the double strand of ORI-T breaks from this side, and the one strand try to pass from F plus to F minus. As you can see, the single strand pass from this conjugation channel from F plus to F minus, right? This mechanism of replication is known as rolling circle replication. Okay, so this rolling circle replication method transfer this one strand of F factor into the recipient cell. At the time of transfer, from ORI-T, the replication procedure also started. So this particular F minus cell will get a single copy of F factor, and here also it is also having a single copy. So due to the replication, it converts into double copy. Here also it converts into the double copy, and ultimately both the cell will get single copy of F factor. As you can see it over, this is F factor, this is also F factor, double stranded. So both the cell become F positive. Suppose you have a tube, just focus, you have a tube in which you have a F plus cell and F minus cell. So when you mix these two cells together, after certain hours of conjugation, you will get only F plus kind of cells here. Okay, so gene from this F plus is able to transfer from F plus to F minus, and both the cells become F positive. Clear? So this is known as F plus F minus mating. Now what is the importance of this? So this leads to the transfer of genes. Gene transfer. Here only genes present on this F factor are able to transfer from this to this. No need or no integration is there. So bacterial genetic material transfer is not possible in F plus F minus mating. Now, another is known as HFR. As you can see here, HFR F minus mating. I will discuss this part along with your topic mapping genes by interrupted mating. Yes. All of you are well? જાગો છો બધા?
Yes, sir. We are all alive and we are enjoying your lecture.
Okay,
Please.
great. So we are discussing about conjugation. We already discussed about the F plus F minus mating. Now this is the 4-mark question theory. For NET purpose, this is known as the 4-mark question theory, because genetic mapping is a very important topic to understand, and definitely one question every time they ask one question from this particular topic, right? So what happened at the time of HFR? I already told you about F plus F minus mating. So in F plus F minus mating, the bacterial chromosome transfer is not possible. But in certain case, what happened? Your F factor and your E. coli chromosome integrates at particular one side. This is that insertion sequence between two genes, valine and threonine. As you can see, valine and threonine, you will find a small sequence which is complementary, and which this is your crossover, double crossover, right? Due to this double crossover, what happened? You will see the integrated F factor. So between valine and threonine, you can see the part of your F factor along with the direction also, right? So this is your HFR. So when this type of HFR transfer from one cell to another cell, what happen? Because for transfer, કોઈ પણ એક જગ્યાથી કટ તો થશે જ. This cutting of F factor from bacterial chromosome sometimes leads to the transfer of these genes in F minus. I will explain this part in the overall animated part in next slide, so it will be easy for you to understand, right? So why HFR important? Because it directly linked with the chromosomal part of bacteria, and this chromosomal part when involves during this conjugation procedure means there are a lot of probability of transfer of bacterial genes into the F minus. Clear? So now I'll talk about directly the HFR F minus mating with context to interrupted mating, right? So this conjugation basically, so several scientists work on this conjugation procedure between the HFR strain and the F strain. As you can see that many of the scientists use the strain called as HFRH. You will definitely find this in many books. HFRH. This H stands for the William Hayes. That is because that scientist isolates this HFRH first. So the most common species used for this purpose is HFRH. But in 1957, Woolman and Jacob, these two scientists from Pasteur Institute in Paris, discovered something new, and they used HFRH cells and made that cells with F minus. Now the question is how this procedure occurs. So everything starts in the flask where you have to mix HFR strain with your F minus strain, and you have to allow these two strains for conjugation. Okay, so don't disturb these two strains, keep it in your incubator and allow it for conjugation purpose, right? And meanwhile that I'll explain this. HFRH cells is having a genotype of THR plus. Now see, કેવા પ્રકારની strain એ લોકોએ select કરી હતી? કોણે? Jacob and Woolman. They selected the HFR strain which is able to utilize 309. That strain is able to utilize leucine, both the amino acids, right? Apart from that, that particular strain is able, it is a resistance against sodium azide, against bacteriophage T1, and able to utilize lactose and galactose, okay, and sensitive against streptomycin. You have to learn how to explain this part. THR positive means that strain is able to produce 309. Leucine positive able to produce leucine. So both the amino acid is produced by that particular strain. But that strain is sensitive against sodium azide, bacteriophage T1 and streptomycin antibiotic, but able to degrade lactose and galactose. And what are the characteristics of F minus? Exactly opposite of it. 309 negative, leucine negative, azide resistance, turn resistance, lac negative, gal negative, and streptomycin resistance. So both the species is having their own characteristics. One is having characteristics of production of amino acids, or as well as one species is having ability to degrade lactose and galactose, and so use that as a source of energy, but are sensitive towards this antibiotics or chemical or bacteriophage. But F minus is having resistance against that. Now suppose this F minus will get genes from HFRH cells, so this becomes maybe 309 નું gene આવી જાય તો 309 plus, leucine આવી જાય તો leucine plus. Okay, so this is how gene transfer helps this F minus strain. We'll see it one by one. So AZI S and R, that is the allelic form. One is for sensitive one, another one is for resistant one. Same as for TON, same as for streptomycin. Okay, so what they did an experiment in which they mix HFR and F minus in a flask, okay, and observe what happened to the F minus recipient cell. So here HFRH cell is your donor cell and F minus is your recipient cell. So here you are interested in characteristics of recipient only. Okay, so in general mixing કર્યા પછી તમારે suppose this is the flask. First I will explain the common mechanism in easy language. Yeah, this flask is having the mixture of HFR and F minus. Okay, so 0 minute, mix it. After 5 minutes, what you have to do is collect a few ml of this and agitate it. Collect it and agitate very hardly. Agitate કરશો એટલે શું થશે કે જે conjugation tube બની છે એ તૂટી જશે, right? This will break. So only few portion of the gene is transferred, not all. So check at 5 minutes, 10 minutes, 15 minutes, likewise this. So at which time, which part of the gene is transferred, you can check. How you can check? After 5 minutes, just cultivate this culture on a minimal plate along with without 309. Don't add 309 in the plate. Just inoculate and incubate this plate, spread it with this 5-minute collected sample and just see what happened. Now just try to recall F minus. This F minus is 309 negative. And our main focus is on F minus frame. Suppose after 5 minutes, this 309 gene is able to transfer from this to this. Means what? This F minus will get this 309 gene and this become 309 plus. So that recipient is able to grow over here. So likewise this, you have to use different type of minimal mediums along with the 309, along with you can add leucine, you can add streptomycin, you can add sodium azide. So you have to prepare number of plates for each minute. Prepare a number of plates, add your sample in each and every plate and find out which kind of recipient were there. You have to check this. Mind it well, all these HFR H cells are sensitive towards this. So streptomycin નાખશો તો HFR grew નહીં થાય, right? એટલે કોઈ પણ streptomycin, sodium azide or that bacteriophage T1, so this donor cell will not able to grow, only recipient. And this recipient is having gene for the resistance, so only recipient is able to grow here. So by performing these whole sets of incubation, you will come across one conclusion. So by doing this, you will know the sequence of gene. Suppose 309 gene pelayo, then leucine, then AZI. Means you can map the sequence along with the time, 5 minutes, 12 minutes, 17 minutes. So based on the minute, you can check the location of your gene on the whole chromosome. It is not possible to transfer the whole F factor along with the whole chromosome. That transfer is not possible. But the experiment suggests that it requires 100 minutes, right? If we talk about E. coli, so in E. coli, 100 minutes is sufficient to transfer the whole set from HFR through F minus. Now see here. So this is known as interrupted gene mapping. Why it is known as interrupted? Because every time, every few minutes, you interrupt the conjugation procedure, right? So 0 minute, 1 minute, 2 minute, 3 minute, you can do. Jacob got up to 8 minutes, no gene were transferred. What these scientists got the data, they got that up to 8 minutes, no genes were transferred. But after 8.5 minutes, the first gene, that is 309, transfers from HFR to F cell. Now see one by one this procedure. So this is your F factor, which is integrated with the bacterial chromosome. Here are the genes, and this is your F minus with the genes. Now mix it at 0 minute. Now up to 8 minutes, they will not get any of this gene transfer. But after 8.5 minutes, they got the gene transfer. So just see, after 9 minutes, threonine, leucine, and that azide gene transferred from HFR to H. After 11 minutes, TON is also there. TON is present is here. So all this is the linear fashion of transfer. Rolling circle model is a linear fashion, and the location of gene is fixed, right? So the sequence remains in a linear form. Suppose F factor 309 થી તૂટે છે એની જગ્યાએ suppose બીજા કોઈ પ્લેસ પરથી બ્રેક થાય. Suppose leucine થી સ્ટાર્ટ થાય છે. So leucine azithrotone remains same. Suppose AZI થી કટ થાય છે. Then AZI to tone to lag. This remains same. The distance always remains same in F factor conjugates with chromosome. This remains same. So you can map it by interrupted gene mapping. Clear? So after 18 minutes up to lag, and after 25 minutes, all the genes mentioned here are able to transfer except streptomycin. All the genes are able to transfer from HFR to H. And these genes later integrates with this chromosome and provides the specific phenotypic characteristics to this F minus. So this F minus become ultimately HFR only. And so this HFR will have certain characteristics, and all the characteristics you will check by putting individual plates, minimal media along with the source of energy, source of essential metabolites or drug. Just try to correlate with the mutant that I discussed in fundamental part. Okay. So based on this, okay, because of this writing, you are not able to see the part, but I have another. These are the interpretation of this interrupted gene mapping. So 9 minutes, the first gene transfer is that is AZI. 11 minutes, that is T1. 18, that is lactose, and 25 galactose. So based on this, you can map the sequence of your gene along with the time. Okay. So in your NET examination, they people will ask you to find out the sequence of genes based on the data provided. I will share that with you. Now this is the complete genetic map of E. coli. Just start from the 309. As you can see, there are various genes along with the time. If you see here, a map distance of 1 minute, 0 to 1 minute, this one, then 2 minutes, 3, 4, 5, 6, 7, 8, 9, 10, 10 minutes, right? So this 1 minute is corresponding the length of this chromosomal segment transferred in 1 minute. એટલે કે 1 મિનિટમાં કેટલું ટ્રાન્સફર થાય છે, that is a 1 minute. If you see the genetic map of E. coli, everywhere you will find the 100 minutes because it requires complete transfer within 100 minutes, right? That it requires 100 minutes, and it always starts from 309. I know we had just 2 hours because it requires at least 2 hours discussion to understand the whole genetic map in detail. But I hope you people are clear how to map the genes using conjugation methodology, right? I will discuss this part in the problem section, so that will be easy for you to understand. So if you just focus here, here you have one problem. Means suppose you have identified a mutant E. coli strain that cannot synthesize the amino acid tryptophan. Means you have a strain of E. coli which is not able to produce amino acid tryptophan, and your task is to determine the location of the tryptophan based on the sequence given over here. So because we have a less time, so without wasting much more time, what I will do is I'm just discussing the few parts with you, facts and the concept. I will provide this part to Nakar sir, so you just go through this part so you can easily understand the mapping theory, just facts of this, because we have only 9 minutes and I want to solve that problems with you all. So directly I will go for the whiteboard explanation, right? So just hold on. So I will share that whiteboard with you. Hope you will get something new in this presentation, or this will be easy for you to understand. I try my best to make it easy for you. So directly we'll discuss the whiteboard. Nakar sir, are you able to see this whiteboard? Actually,
Yes, sir.
this
Yes,
is the
sir.
blackboard. No, this is not the whiteboard.
Blackboard.
This is the blackboard actually, right? So which kind of questions were asked in a NET examination? Just go through this. I'll share some problems with all of you. If you focus here, The first question is the co-transduction experiment was performed to decipher the linear order of 4 genes. So here, first, how to solve this kind of questions? So first, read it very properly. What basically they want to explain. So here, they discuss about the one co-transduction experiment. We'll discuss transduction in the next part, but this is the transduction experiment where they perform the just hold on, this is not working. Don't know what happened to this. Okay, now this is okay. So here they are discussing about 4 genes. All genes are A, B, C, and D. And 3 sets of the experiments were done. First set, second set, third set, right? And they got certain kind of results. Result in the form of frequency. Means what? If one gene, that is A plus, and they check the transduction of A with B, A with C, and A with D. After this, just grow this on plate. And after growing this, after growing this on the plate, what you have to do is you have to find the number of colonies appear. Right? So A plus B, A plus B plus, A plus C plus, A plus D plus. So for A plus and B plus, you got 31 colonies. For A plus and C plus, 3 colonies. A plus and D plus, that is 89 colonies, right? So these are the co-transduction experiment and the frequency of colonies on the plate. Now, what is your task? Based on this frequency shown here, you have to identify the most likely order of the gene. You have a 4 gene here, A, B, C, and D. So, is it correct? Or B, C, D, A. Likewise, this, you have to find out the sequence of gene. So, how you find out the sequence? I'll give you the very simple explanation and the easiest technique, right? Means if A and B are nearer to each other, you will get the maximum colony. Here, you just see A and B 31. The maximum is 89. It indicates that A and D both are nearer to each other. Then A and B, then A and C, right? So here I'll change the color of the pen. So, okay. So here A and D nearer to each other, right? Now that if they are nearer to each other, then you will get this maximum frequency because of this co-transduction experiment. Now, if you see the set 2, maximum frequency is here. That is B and D. And the third one, maximum is this one. So C and B. Now, just put it here. A. એની સાથે સૌથી નજીક જો તો કયું છે? That is D. Now D. Which gene is nearer to this D? Gene B is nearer to this D. B and C. Now this is the sequence of your genome. Now find out A B C D, B C D A, C D A B, and A D B C. A D B C is here. So your right answer is ACBC. So at the time of the examination, don't go for this kind of calculation. A time bagadse. Just try to focus on the frequency. Based on the frequency, find out the nearest gene and club your nearest gene data, and you will find the exact sequence of that gene. Right? So because these kind of questions were already asked in 4 marks question, and that requires a lot of time. So by doing these simple tricks, you can save your time. Now this is the another question. Sir, do we have actually it is 6 o'clock. So what about the time, sir?
Yes, sir. Just 1 minute you can conclude. Next time we will arrange all the other
So
things.
what I will do, sir?
Yes.
I will continue all this in my next lecture, and I will share this whiteboard details with you, so you can share these details with student also for the practice
Yes.
purpose. So these are the main 5 types of questions asked in NET examination, રાઈટ. એટલે છોકરાઓ
Yes, sir.
ને આ 5 પ્રકારના ક્વેશ્ચન્સ જ ઓલમોસ્ટ આવશે, એનાથી બહારના નથી આવવાના. આ જ 5 ક્વેશ્ચન્સ અલગ અલગ રીતે પૂછાશે. ધીઝ આર ધી concluded types of questions asked in NET examination. So what I will do, I will share this file with you, so you can share with all the students so they can practice by themselves. So in the next lecture, I will start with the calculation of this. So we'll provide them as a homework if they want to perform by themselves. Clear?
Sure, sir.
Because of the time limitation, so if I will conclude this part, actually this is not the conclusion, but this is just the hold.
Yes, sir.
Right, because genetics is a quite huge topic to finish, just like immunology. So we'll continue with this problem part in the next session. And before that, I just gave you the few nice books for your preparation purpose. You can start with the basic book, that is the Prescott, because it is having a very easy language for Gujarati medium students. Then just go for the Snapstead and the Simons, that is the principle of genetics. This is really nice book for NET preparation. And if you want some good language, if you are now comfortable with basic English, then you can go for this modern microbial genetics book. So all these books are available on NET, no need to purchase. Nowadays during the lockdowns, many books are available for the students, right? You can follow me, definitely you can follow. I'm a technology lover, so you can definitely find me at all these social media platforms. I have my own YouTube channel. You can find many videos regarding the microbiology on the YouTube channel, or else you can ping me on Instagram. If you have any query, definitely you can ping me on Instagram. So at the end, I will not conclude this session because I want it to be continue. So in this session, I can say that we covered the fundamental part, and definitely for your preparation, this lecture will help you a lot. Along with this, we discussed the transformation in pneumococci, the basic principle given by Griffith, and Bacillus subtilis, the mechanism in Bacillus subtilis. Definitely you will get questions from that part. And we discuss about the basics of conjugation and the interrupted gene mating. I know I'm unable to complete that problem part of interrupted gene mapping in this lecture, but definitely I will discuss that part. I will explain it in a very calm manner in the next lecture, right? Because we have time limitation. But thank you so much for inviting me, sir. I tried my best to concise everything in a single presentation. Hope you like this. Hope the students will like the video. Definitely, your feedbacks are energetic molecules like ATP for me. So definitely, students, give your response or give your feedback. I want criticism also. So if you have a negative feedback, that is the positive side for me. So definitely, without any hesitation, please do the feedback for this presentation. Anything you want, you can call Nakar sir, as well as I gave you the all the platforms. You can search me in Google as a Dr. Dehin Bhagat. You will definitely get the all 14 hits of mine. So my contact is very easy because you can easily contact me on Google. You can find my WhatsApp number on Google. So anytime if you have any query regarding the science only, I will always ready to help. Thank you so much, the team of SDGU, for conducting this kind of knowledge sharing sessions for the students, specifically students from Godhra, that territory. Basically, I'm from that part, so I know the condition over there and the exposures for the students. So you did a phenomenal job, sir. I'm really thankful to all of you and the whole team. Thank you so much.
Thank you.
It's up to you, sir.
Ji, thank you so much, Dehin sir, for delivering such a nice, detailed, and comprehensive lecture on fundamentals of genetics of microbes. And now, for vote of thanks, I would like to invite Falguni madam to go for the process of vote of thanks. Your voice is not coming, Falguni madam. Still Still it is not coming. No. No. Hello. I think there is some technical issue that we have gone through. No issue. We are thankful to Falguni madam, but we have some technical issues, and I think there is some mic issues. But we can see you, and we can see the feelings that you are having. On behalf of you, I will do the proceedings because we have call from university. It is already 6 o'clock, and they have to leave the office. So we are thankful to our Vice Chancellor, Sir Dr. Pratap Singh Chauhan. We are thankful to Dr. Anil Solanki, sir, who is in charge registrar, sir. We are thankful to Ajay Soni, sir, for providing this stage and opportunity to all of us. And in addition to that, we are thankful to today's esteemed speaker, Dr. Dehin Bhagat, who delivered very nice and extensive lecture. We are also thankful to our both the students, particularly Vijay Bharwad, who joined us, and he left us quickly due to some technical problems. And we are also thankful to all of us who are watching us daily whenever our lecture is organized on YouTube and Facebook. And we in the end, last but not least, we are thankful to the arranger of this program on social media platform, Jagrut Bhai, and the team of Sri Govind Guru University for the benefit of students. So with that, we conclude our meeting, and we really enjoyed the lecture, Dehin sir, and definitely we will enjoy your lecture as well in the future also. So with that, I think I should conclude the meeting over here, and still till then,